So our next speaker is PRASIFKA, who is at the USDA-ARS in Fargo, where he is a research scientist.
Thanks, Ben. So I'm the lone entomologist in the sunflower and plant biology group here in Fargo. And I think the image on this first slide is familiar to a lot of you, but maybe not to everybody. This is a hybrid seed production field in North America. This primarily happens out west in the Sacramento Valley in California. In the middle here, you see multi-headed male lines, and then surrounding them, single-headed females that have cytoplasmic male sterility. So this is what it looks like here when hybrid seed is produced. And I'll just admit, I don't know everybody on the call. Some of you may know more about bees than I do. Even though I'm an entomologist, my background is more in biological control and integrated pest management. And the position I was hired into at USDA over 10 years ago was designed for me to work on host plant resistance, that is helping breed sunflowers that are resistant to the insect pests that we have. So little things like this red seed weevil that's really our #1 seed feeding pest. But in interacting with our stakeholders, including a lot of growers in the area, they seemed to indicate that they knew how to kill pests, but they did not know how to make bees do what they wanted. And so because of my own curiosity and because of urging from those folks, from stakeholders, we started working on bees several years ago with sort of two basic goals. One, to understand bee foraging and sunflower, and secondly, to improve pollination, and if we can, pollinator health. And so, Stan's talk earlier talked a lot about improving pollination, and I think that collectively we've made some progress on that. The pollinator health part of it is more complicated, and certainly I will admit to not having made much progress in that area. So the slides that follow this introduction are largely summarized by a paper that we published a few years back. Some of the results from today are a little more current, but really it was kind of an explanation of what we're trying to do. Several years ago, there was a paper published about pollinator dependence of agricultural crops. And so they sort of characterized, this is what sunflower is like, this is what soybean is like. And sunflower was listed as having a low to moderate dependence on pollinators. And that always struck me as kind of curious. So in terms of companies that produce hybrid seed, because we have cytoplasmic male sterility, we have this 3-line system with A, B, and R lines. We're 100% reliant on pollinators to produce the seeds that growers plant. So that's part of my argument against this low to moderate dependence. And then again, Stan mentioned this, that even for hybrids that are bred to be self-fertile, we get sort of wildly different estimates. Some hybrids seem to do really well without bees, and other hybrids seem very reliant on bees to produce a reasonable yield. And even those ones that are the very best, when you look at them under varying environmental conditions, even those self-fertile hybrids need bees. The thing that other folks have implicated is that this is related to stress in and around bloom. That appears to be important in taking a self-fertile hybrid and sort of increasing its pollinator dependence. We know from previous research that bees, particularly in North America, wild bees, show really fine scale preferences among sunflower lines. So just planting a couple of inbreds or hybrids next to each other, you can really clearly see their preferences. And in lots of other crops, and I sort of cut the table off, but a lot of crops, these preferences are at least significantly explained by floral rewards. And so I think that's a reasonable place. That was a reasonable place to me to start in sunflowers. So these things sort of combine to suggest to me that there's potential to breed the crop to be better for bees. We have variation, clearly heritable variation in key traits that govern pollinator preference. We have a sunflower genome available to us to enable sort of marker-assisted breeding. And really, I'll argue sort of the complicated part is to figure out how to do it, how to create a plant that's better in terms of its agronomics and maybe also better for bees. And I'll just admit, even if we do sort of great science, it's not clear whether this will change anything. Sunflower growers understand that bees are important. And we do these big crop surveys every couple of years where primarily we're looking for collecting pest data. But we see the achenes that don't get filled in these grower fields when we're x-raying for pest damage, we see empty achenes. So we know and they know that full pollination is desirable and useful, but it's a difficult thing if you're thinking about planting decisions, if you're thinking about crop management, particularly with insecticides. I think it's an open question whether, even with really good research, whether we can succeed in trying to do what I'd like to do in sunflowers. So the objectives that I'll talk about, and really what we've been doing for the most part over the last several years with regard to pollination, is first looking at benefits of open pollination for current hybrids. So we do have estimates from previous research, but breeding for self-fertility is ongoing by both public and private breeding operations. So I think it's useful to take a snapshot of that every so often. We also want to associate bee preference with specific crop traits. And again, there's previous research that talks about lots of different traits, some of which are really easily measurable and some of which are a bit more nebulous and hard to wrap your hands around. And then the last part is trying to put it all together to some degree, using the genetics tools available to map traits of interest and really figure out an approach to benefit the crop and benefit pollinators. And again, I'll just admit, that's the complicated part that still needs a lot of work. So these are some trials over about a 4-year period, both in North Dakota and then later expanded to South Dakota and Nebraska. And these are all central United States where most of our sunflowers are produced. North Dakota specific trials, we had 15 confection hybrids, 5 hybrids from each of 3 seed companies. The range in terms of benefits from open pollination, and again, we're using the same kind of exclusion bags that Stan showed. The range of value on a per hybrid basis was 0 to 108% increase in yield. And you can see some of the summaries for the multi-state trials over here. More pollinator dependence in 2016 and more in Nebraska than these other sites. But 26% yield increases in North Dakota under open pollination across all those hybrids. About 45% increase in yields when we included 3 states over 2 different years. And it's important to note that the amount a given hybrid benefited from open pollination was correlated to how attractive it was to bees. And in this case, the bees are these wild solitary bees that we have in the genera Melisodes and Andrena. Very, very, very few honeybees in these fields. I would say in many cases, they'd be less than 2% of the bee visits. It's worth remembering that not all bees are equal in terms of their pollination efficiency. And these are some data that were taken by having heads that were bagged all during pollination except for a single bee visit. And so you let one bee onto the head and then shoo away any additional bees. And there are differences among different types of bees in how long their visits are. But even these Melisodes, males and females, their visits are very similar in duration, but the females are much better pollinators in terms of the number of seeds produced by a single visit. And so these are CMS heads and only able to produce seed by that one bee visiting and foraging on the head. So I mentioned before, hybrid seed production is totally bee dependent, and the data from the previous slide over 4 years and 3 states, it's roughly a third, a 33% increase in yields in the central United States. And whether that seems large or not, I'll just note that that's probably taking a farmer from growing sunflowers at a loss to making a profit. Those data were from confection sunflower hybrids. We haven't had any data from oilseed sunflowers that was published in North America in something like 15 years. But we have reason to believe that their pollinator dependence is not that different from confections. I mentioned before that in these locations in the central United States, we don't get many honeybee visits. And I'll just draw your attention to this map of North Dakota on the right. And all those blue dots are honeybee colonies that are placed out on the landscape. So North and South Dakota are the 2 primary sunflower producing states in North America, in the United States. And they're also the 2 primary locations where honeybees are over summered. So all those blue dots are honeybee colonies, and our field trials are over here at the edge of where those blue dots become really abundant. So in our case, it's kind of a preference issue, not a presence of honeybees in the area. I have some concerns about how the data that are generated from these trials are interpreted. And this is, again, relevant to some of what Stan presented. We know that bagging is an imperfect solution to do pollinator exclusion. And I think it's because the bags will change the environment around the head, change the temperature and humidity. Those Delnet kind of bags are better than cloth bags, but we don't have a perfect solution for how to exclude pollinators. We also see some folks that are offering products or pollination services to our growers that I think I can just charitably say are unproven technologies. And I think that they use these kind of data to sell their product to growers without any evidence of the efficacy of their product. Changing gears from how important are bees in the crop, but over to how bees make decisions related to foraging. I could have a long list of previous studies that looked at various plant traits associated with pollinator preference of sunflower, but I've just broken them into general categories, things like visual appearance, odor, and then these floral resources, nectar and pollen availability or quantity and quality. And this is work where we were looking at 20 inbred lines. They were A-B line pairs, meaning the same genetic background, but those A lines had cytoplasmic male sterility. And we're using sort of traditional methods to assess nectar volume with microcapillaries and looking at nectar concentration using a refractometer. And so looking at those released inbred lines in their A-B pairs, we found support for primarily 3 traits. The presence of pollen was important to wild bees. The amount of calories available from nectar is important. And I've underlined floret size, and that's what I'm going to focus on in some of these subsequent slides, and I think you'll see why we're focusing on that. So, floret size is easy to measure in the sense that if you're pulling florets off of a plant and off of plants in sort of a regimented way, standardized location and standardized maturity of the florets, you can put those florets on a scanner and measure them with software like ImageJ, and it's not influenced by the observer if you do it right. We also knew that in unrelated plant species, folks have been successful in mapping the genes that govern floret size. So we thought that we would have some luck with this. And I'll just mention, again, here we are in the United States, in the continental or contiguous United States. Here's North Dakota, and we'll hop down here to Arizona, a much more arid environment for a couple of slides. And here you can see the continuum of floret sizes available in public germplasm, essentially bee-line germplasm. And going from here to here is about a 1 millimeter change in floret length, 2 millimeters, 3 millimeters, and a dime for context scale. So this was a trial with, I think, 30 inbred lines in North Dakota several years ago, and we're looking at the association between floret length and visits by these wild bees, Melissodes andrina. And out of about 7,300 bees noted on these plants, about 2% were these really large-bodied bumblebees, but the majority were small and medium-bodied bees. And just looking at the length of florets explains more than half of the variation in wild bee foraging on these inbred lines. So that's really kind of felt like a big deal at the time that it could be that simple. It has nothing to do with how much pollen they produced or nectar produced. It's just accessibility. And I have my little bee with a cartoon paper straw, which growers, it helps illustrate why floret size is important. Simply, it's how easy it is to get to that nectar reward. So the previous slide was really not relevant to honeybees. And one of the things we wanted to do was know whether or not the same relationship applied to honeybees, because they are important in the United States. When you go out into the Sacramento Valley and you have hybrid seed production, the companies that produce seed out there do bring in honeybee colonies to ensure high yields in their seed production. And the data aren't quite as tidy, but really this work in Arizona supported the idea that the same trait, the floret length or depth, if you like, is important in explaining honeybee foraging, just like it is these wild bee species. You can see pollen on some of these bees, it's clear that they weren't all simply nectar foragers, and that's relevant to a subsequent slide. Another thing that we've looked at is what happens to the diversity and the community composition of bees in response to variation in floret size. And so this was work done back in North Dakota again a couple of years ago. We did it for 2 years, and the results were sort of qualitatively similar between years. Looking at individual lines of sunflower planted in different plots, the diversity of bees didn't really change. We had 5 or 6 bee species that were collected in each row, but which bee species were found on an inbred line did differ between lines that had small florets, medium florets, or large florets. And you can see, looking at bees that were collected in these individual plots, a little bit of a difference. This is for early planted sunflowers. When you go to the next slide, it's a bit more dramatic. And so these were blooming 2 or 3 weeks later. And that late sunflower bloom period is when these large-bodied bumblebees can show up in our field trials. And so this is a lot more noticeable difference. than for those early planted sunflowers. So we do think that planting time and flowering time are an important thing, at least where we are, to keep in mind for these trials. I mentioned we'd really briefly come back to the work in Arizona. Because we saw so many pollen foraging honeybees, we thought we'd outsmart them, and we failed. So we planted the male sterile equivalent in the subsequent year, and we thought, that would be great. And really, the results changed a lot. The overall bee numbers were poorer because of the absence of pollen. And also the cytoplasmic male sterility does some weird things to other aspects of floral biology. So we really didn't see those responses to floret depth like we expected. We saw some response to nectar, but it seemed like it was a 0-1 in that lots of our lines produced almost no nectar, these CMS lines in that environment. It was really viscous and really difficult to sample successfully. So that was a reminder of how much is yet to learn. I think that even though I listed those 6 traits and then said we thought 3 were important, and then I really focused on 1, they're probably all important under some contexts. I think the environment is important. I think it's always good to provide easy to access nectar, that all things being equal, these short florets are probably almost always good, unless you're solely reliant on big body bees like bumblebees. There are other things that we don't know. What about qualitative variation and reward? So what about the nutritional content of pollen or details in regard to nectar composition beyond just how much sugar is there? And I don't think we understand well what foraging bees are using. Bees are capable of doing all kinds of crazy things, like looking at the electromagnetic field around the heads. And that's something that we have not tried to get into yet. Going to the last subject area for these objectives, it's clear that having a sunflower genome available should enable marker-assisted selection, marker-assisted breeding, and we know that that's being used in commercial breeding programs and also in public research programs like INRA and USDA. And it's hard many times to know what our corporate stakeholders are doing, but if they use traits that are released in public germplasm in their material, there is sort of potential to transfer improved pollinator attractiveness in a package with other traits. So we don't know what they'll do, but we do know that they use the public germplasm that gets released, and so that's one way to get these traits used. So what are some of the challenges to doing what I've proposed? I think that quality mapping of some of these traits is really hard. And so, calling up some folks before we started trying to do some of the nectar sampling, they just essentially said that it was crazy to even attempt it. And I don't think it's crazy, it's just really hard. I think it's also fair to acknowledge that there are lots of other breeding needs that have nothing to do with bees. And so it's difficult to ask a breeder to add one more thing to their workload in terms of trying to select for many, many different traits. The potential benefits of succeeding at something like this, I think you can improve yields, but I think primarily what you would improve is yield stability, meaning in those years where the environment is difficult, whether that's heat or drought or too much moisture, having pollinators helps stabilize your yield. I think we'd like to improve forage for bees to benefit both managed bees and wild bees. And whether this is really successful in sunflower, I think there are probably other crops, higher valued crops, things like blueberries or cranberries, that are really good chances to do this kind of work successfully. So this is a distillation of a lot of work looking at about 240 inbred lines for floret length. And floret length in this case is a proxy for nectar accessibility. How easy and efficiently can these pollinators get the reward? These little circled areas are locations on the sunflower genome that are that have QTL for the length or depth of those florets. And in 3 out of the 4 cases, those QTL are not near QTL that govern seed size, which is also an important trait. So really, it seems like we should have the ability to hold seed size constant and shrink floret size using marker-assisted breeding. And we're trying to do the same thing with regard to the nectar rewards. So there's a mapping population. In this case, it's a biparental population where we crossed a high nectar producer with a low nectar producer and did the phenotyping under controlled conditions. And that was necessary both to remove environmental variation, but also doing this in a growth chamber, you can increase nectar differences relative to what you see in the field. Nectar production was correlated with the sweetness of the nectar, and we did validate these growth chamber results. This is a histogram showing one's like the low parent and one's like the high parent. And the mapping is still being completed, but it really looks like 1 or 2 QTL and possibly a modifier. Hopefully that's something we can complete and submit here in the fall for publication. So we really don't quite have an ideal understanding of what bees experience in the field. As I mentioned, these lines that are really strongly contrasting in a growth chamber where the nectar ratio is like 8:1 between a high nectar producer and a low nectar producer. When you put those out in the field, it's closer to 2:1 in terms of calories available. We often use the pollinator exclusion, and I think that artificially increases the nectar resources if you're unbagging those plants for a period of time in the day. And so we know that that's a bit unrealistic. So I think there's some work to do looking over time with bees constantly removing nectar and pollen to understand how they make their foraging decisions. We need to understand what the benefits are to bees of sunflowers that offer greater or lesser resources. And I think we also need to consider whether improving sunflowers functionally adds to the risk of insecticide exposure for bees. And so we really have some devastating pests in North America. This is a head that has been mostly consumed by larvae of the sunflower moth. Even prior to bloom, they've eaten up most of the florets. So it is something that we need to think about. And with that, I'll try to summarize the work. We know the bees are essential for sunflower profitability, and that's true for the seed companies making hybrid seed, and that's certainly also true for farmers growing those hybrids. We know that there's genetic variation for bee preference to inbreds and hybrids, and that's clearly linked to floral resources, most strongly to the availability of nectar resources in our plots in North America. And it's positive that we see some success in our ability to map these nectar-related traits. Even though in some cases, looking at things like composition of the nectar, we don't know what the value is yet to the pollinators, but we do know that we can control those traits rather simply. So as I alluded to earlier, I think we don't understand what nectar and pollen quality mean for the bees, both in terms of preference and their health. And we have some difficulty predicting foraging of the bees in different environments. Really, the open question is, if we understand what bees prefer in this crop, and we understand the genetic basis for that preference, what do we do with that? Do we develop and release new inbred lines? Do we further demonstrate the value of bees to stakeholders and hope they'll express their preferences to the companies that they buy seed from? Do we try to take our work into other systems? Really, I and collaborators, folks like Brent Holke here in Fargo, are open to suggestions about how we do those things, how we maximize the utility of the work. And with that, I'll stop to acknowledge some of the collaborators. I mentioned Brent. Rachel Mallinger was a former postdoc in the lab. Beth Ferguson is a current postdoc, and we had a previous technician, postdoc, and undergrad that helped with a lot of this work. And yeah, when we get to the break, I'll be happy to answer questions.
All right, wonderful. Thanks so much for the great talk.